In this lecture, we will look at innate immune responses. You will remember that the innate immune system is the first line of defense, physical, chemical, and cellular mechanisms mounted against invaders. We will look at innate responses to viruses by looking at the human immune response to hepatitis C virus in detail, bacteria, and we'll look at the example of Treponema pallidum that causes syphilis, fungi with the example of Candida albicans, and parasites, and we will take the example of Plasmodium falciparum that causes malaria. Then we will look at a controversial treatment for syphilis developed in the 19th century by Julius Wagner Jaureg called malariotherapy. Jaureg won the Nobel Prize in 1927 for this therapy. I would recommend you look at the notes in this website where I discuss the historical context and the ethical aspects of this therapy. Please click on the adjoining link to access the notes. So let's get started with a background about the hepatitis C virus. The hepatitis C virus belongs to the Flaviviridae family of viruses, a family that includes monopartite positive single-stranded RNAs. The genus that HCV belongs to is Hepacivirus. This genus contains 14 species from Hepacivirus A to N that infect various hosts. HCV belongs to the species Hepacivirus C. HCV infection affects 1.6% of the world's population with a prevalence of 0.1 to 23% depending on the region. This map shows the percentage of people infected with HCV in 2015. Out of the 6 major genotypes described for HCV, the most common is genotype 1, which is responsible for 46% of all infections, followed by genotypes 3, 2, 4, and 6. HCV infection is mainly caused by iatrogenic transmission in low and middle income countries and by risky sexual practices and sharing needles while tattooing or injecting drugs in developed countries. As you can see, in US, 60% of HCV infections is caused by drug use. 15% is sexual, 10% is unknown, and the rest of the 15% is iatrogenic transmission. After HCV entry, the virus produces an acute infection which is asymptomatic in 75% of cases and symptomatic in 25% cases. The infection spontaneously resolves in 5 to 20% of cases within the first 12 to 16 weeks. It may, however, become chronic in about 80 to 95% of cases. In many cases, the infection remains undiagnosed for a long period of time, as long as 20 years, as chronic infection is frequently asymptomatic. In 20% people, the chronic infection does not produce disease. Most chronic infections cause a liver injury that may progress to liver fibrosis, cirrhosis, and eventually hepatocellular carcinoma. Therefore, it is important to detect chronic HCV infection before the development of liver disease. Detection methods include identification of HCV RNA by polymerase chain reaction, HCV proteins by ELISA. There is no vaccine for the prevention of HCV infection, but direct antivirals have response rates higher than 90%. Hepatitis C virus is a small 40 to 80 nanometer in size enveloped virus that contains the viral genome bound to the core protein. The viral genome is a single-stranded RNA molecule of 9.6 kilobases and has positive polarity. The genome serves as a messenger RNA and consists of long open reading frame ORF flanked by highly structured 30 and 50 untranslated regions UTRs which are required for viral translation, replication and encapsidation. The 30 and 50 ends of the HCV interact with each other. The 50 UTR includes an internal ribosome entry site, IRES, that allows cap-independent translation of a single polyprotein. The polyprotein is approximately 3,000 amino acids in length and is co- and post-translationally cleaved by viral and cellular proteases to produce all mature viral proteins, 3 structural proteins, core, E1 and E2, and 7 non-structural proteins, NS1 or P7, NS2, NS3, NS4A, NS4B, NS5A, and NS5B. Importantly, HCV proteins are characterized by multifunctionality. The core protein is the main component of the HCV nucleocapsid and promotes nucleocapsid assembly. The envelope of the viral particle contains the viral E1 and E2 glycoproteins required for cell attachment and entry and for assembly of the infectious viral particle. P7 participates in HCV assembly and release. NS2 autoprotease is required for the cleavage of NS2-NS3 and participates in viral assembly. The replicase complex is formed by NS3, NS4A, NS4P, NS5A, and NS5B. NS3 is another viral protease and helicase. As a protease, NS3 processes non-structural proteins and cleaves some host factors involved in the antiviral response. Helicase NS3 is crucial for HCV RNA unwinding and replication. And it is also essential for viral particle assembly. NS4A helps binding of NS3 to the endoplasmic reticulum and increases NS3 activity. NS4B participates in viral assembly and is a key protein for formation of membranous web, MW, required for replication. NS5 binds several proteins, the viral RNA, and the membranes to participate in MW formation, viral replication and assembly, and to block the antiviral response. NS5B is an RNA-dependent RNA polymerase essential for viral replication and assembly. Similar to other RNA polymerases, NS5B lacks proofreading activity. In vitro, hepatocyte and lymphocyte cell lines, especially the HUH7 cells, are especially good for growing this virus. In vivo, the virus attacks hepatocytes. Hepatitis C viral particles circulate in the bloodstream as lipoviral particles attached to lipids and apolipoproteins from the host. The shell facilitates the infection of hepatocytes and may aid in viral escape from neutralizing antibodies. HCV entry into the hepatocytes is a highly regulated multi-step process that involves various cellular proteins. Once the virus is in the liver, it is captured by heparan sulfate proteoglycan receptors present in the basolateral membrane of the hepatocytes. There, LVPs are sequentially hydrolyzed, the associated ApoB is uncovered, and the ApoB exposure enables low-density lipid receptors binding to the LVPs. Then, These LVPs interact with scavenger receptor class B type 1 and CD81, which allows migration of the particle to tight junctions binding to claudin 1 and occludin. They are then internalized by endocytosis. After uncoating driven by the acidification of the endosome, the viral genome is released into the cytosol. Once the HCV genome is in the cytoplasm, viral replication takes place in a specialized structure formed by double membrane vesicles originating from the ER called MWs. NS5A binds the HCV RNAs and the viral RNA polymerase NS5B oversees replication. First, the positive stranded RNA serves as template to produce intermediate negative stranded RNAs. Then the negative stranded RNAs are copied into new positive strand RNA viral genomes, which can be used for translation, replication, and packaging. Hepatitis C virus, a viral particle assembly occurs in the ER close to the membrane-bound lipid droplets. NS5A binds the 30 UTR of positive stranded HCV RNAs and transfers the genome from the replication complexes in the MW to the core proteins bound to the ER. Then the nucleocapsids acquire their envelope, most probably after budding into the ER. The new HCV virions adhere to the nascent VLDL or LDL particles and form the LVPs that are transported to the Golgi complex for viral release. Therefore, a proper functionality of the lipid secretion pathway and the proper synthesis of VLDLs is essential for HCV release. HCV can also be transmitted directly from cell to cell, escaping the inhibitory action of neutralizing antibodies. Let's now turn to the innate cellular responses to HCV. To start with, let's understand the general overview of the cell cascade. HCV PAMPs, HCV pathogen-associated molecular patterns are recognized by inactive RIGI, protein produced by retinoic acid inducible gene, to cause conformational changes in RIGI. RIGI then gets ubiquitinized by TRIM25 and Riplet. This activates RIGI. Active RIGI is recruited by MAMs, mitochondria-associated membranes, and the chaperone protein 1433E to MAVs, mitochondrial antiviral signaling protein, releasing TBK1, IKK epsilon, IKK alpha beta. TBK1 and IKK epsilon phosphorylate interferon regulatory factors IRF3 and IRF7 to stimulate interferon 1 and interferon lambda production. And IKK alpha beta activates NF kappa B, leading to the production of pro-inflammatory cytokines. Interferon 1 and interferon lambda recognized by IFNA receptors and IL-28, IL-10 receptors respectively. This leads to phosphorylation of signal transducers and activation of transcription STAT1 and STAT2. STAT1 and STAT2 and IRF9 form the interferon stimulator gene factor ISGF3 complex. The ISGF3 complex translocates to the nucleus. Tyrosine kinases JAK1, TYK2, and IKK epsilon phosphorylate STAT1, which results in interferon stimulator gene ISG induction. Interferon lambda then activates IKK epsilon. Interferon 1 induces TRIM22. TRIM22 ubiquitinizes viral NS5 and inhibits HCV replication. Interferon 1 also induces ISG15. Isolation of viral NS5 then inhibits HCV replication. Now let's look at this pathway more closely and how the virus can evade this immune response.